Next topic is linkage. Physical association of gene on a chromosome, it is known as linkage. Sex linkage was first discovered by Morgan in D. melanogaster fruit fly and first coined the term linkage by Morgan. Actually, good set level as well as NEET level linkage map questions are there. Fine. The distance between the two genes. Okay. એવી રીતે લિંકેજ મેપ તમને પૂછે છે એના ક્વેશ્ચન્સ અહીં આવશે. In case of linkage in dihybrid experiment, capital A, small a, capital B, small b, here two types of gametes are formed. Capital A, capital B is to capital A, small b. Sorry, small a, small b. Whereas in case of independent assortment. In dihybrid, that is capital A, small a, capital B, small b, same, but here capital A, small b, and here small a, capital B. So here four types of gametes are formed. Which are they? Capital A, capital B is to capital A, small b is to small a, capital B is to small a, small b. And you know that in dihybrid experiment, the recombinant characters are due to independent assortment. New characters are there. Linkage genes are linearly located on the same chromosome. Now, the number of linkage group how we identify the number of linkage groups? Number of homologous chromosome pairs or we say that number of haploid number of chromosomes. Now we draw the one table here. To N pair and linkage group. P plant 14 chromosomes are there, 7 in haploid, in pair 7, and linkage group is also 7. Means 20 chromosomes are there, 10 in haploid, 10 pairs and 10 linkage groups. Drosophila, 8 chromosomes are there, 4 in haploid, 4 in pair, and linkage group are 4. Barley, 14 chromosomes diploid, haploid 7, pair in that 7, and linkage group is also 7. And mouse, 42 chromosomes, 21 pair, in haploid 21, and linkage group also 21. I think you know this. The strength of linkage is inversely proportional to distance between the two genes. In Drosophila, in linkage and crossing over, in cross A, the distance between two genes is less. So linkage characters are more, 98.67, and crossing over ratio 1.31. But see, in the cross B, the phenotype is white color miniature wings, and the distance is more. So in there, linkage characters are less, but crossing over characters are more. So strength of the linkage is inversely proportional to the distance between the two genes. Factors affecting crossing over CO. distance between the two genes increases, crossing over increases. The most important, age increases, crossing over decreases. Fine. So this is the most important one. Somewhere else you heard this MCQ. Fine. Age increases, crossing over decreases. How the sex is affected in male, crossing over decreases. Crossing over is totally absent in male Drosophila. That's why at the time of the topic linkage and recombination in Drosophila, I had told to you, you know that the lifespan of the Drosophila 2 weeks. The lifespan of the Drosophila 2 weeks or 14 days. Female Drosophila is little bit larger than the male. Why Thomas and Morgan select the Drosophila fruit fly? Because it is easily cultivated in the laboratory. And within the short period of time, much more number of offsprings are formed. Okay? So D. melanogaster, it's a unisexual insect and it is easily cultivated in the laboratory and 2 weeks lifespan there. Fine? So these are very important one. So here the distance between the two genes increases, crossing over increases, and one thing if you know that in meiosis division in prophase 1, packaging phase, if you know that in meiosis in prophase 1, in packaging phase, the length of the chromosome is more. Length of the chromosome is more than more number of chiasmata. And this more number of the chiasmata formation leads to more crossing over. So here you also consider the length of the chromosome. If the length of the chromosome is more crossing over, because only in meiosis cell division, fine, only in meiosis cell division crossing over occurs. And when the chromosome is twined around each other, at that time the number of the chiasmata formation more. That's when more crossing over. So here the distance between the two genes, here you consider that, but along with that you consider the length of the chromosome also. So if you think about, you connected the human genome with this topic. First chromosome is longest one. First chromosome is longest, and X chromosome in female, it is smallest, and in human being The Y chromosome is smallest one. I think if you know that in first chromosome 2968 genes are there. In first chromosome 2968 genes are there, and in Y chromosome 231 genes are there. Okay, that is given in the Human Genome Project. What is cis arrangement? Cis arrangement means both the dominant genes are present on one chromosome and both recessive genes are present on one chromosome. This is known as cis arrangement, and this cis arrangement is original arrangement. Here two types of gametes are formed, capital A, capital B, and small a, small b. Now second topic, transarrangement. transarrangement. Transarrangement means one dominant gene and one recessive gene present on the one chromosome and opposite side on one chromosome. The dominant gene's recessive gene is present. It contrasting trait. And recessive gene's dominant gene is present on the other side. It is not original arrangement. It is actually due to the crossing over, and two types of gametes are formed. Okay, so here you properly clear the topic. Independent assortment, due to that four gametes are formed, and due to transarrangement 2 gametes are only formed. Okay? No any type of the parental characters are there. Now the types of linkage. First is complete linkage. Complete linkage. Here, complete linkage always show parental combination. No chance of the new combination. No chance of crossing over. Here, the two genes are very close. Two genes are very close to each other on chromosome. It is very rare in nature. Majority in some organisms only. We identify the complete linkage. Just for example, in male Drosophila. Yes, previously I told to you that no chance of crossing over in male Drosophila. So this indicates majority parental characters. Along with that, similarly in the female silk moth, you know that silk moth cultivation known as sericulture, right? Second is incomplete linkage. Incomplete linkage. Less than 50% new combinations are formed along with the parental combination. So little bit change. 50% characters are of the parents and less than 50% characters are new. Here new combinations are formed due to the crossing over. Right? So this is known as incomplete linkage. Incomplete linkage is mostly seen in newly formed offspring. Okay? Now, application of linkage. I have given you the one equation in linkage and recombination topic. Same. Application of linkage. Here, the strength of the linkage is inversely proportional to the distance between the two genes. There I have written that, but it is inversely proportional to crossing over also. So the unit is centimorgan, cm. Right? Centimorgan. Here C is small, M is capital. Now the sex linkage. When genes are present on the sex chromosomes, this type of the genes are known as sex-linked genes, and such a phenomenon is known as sex linkage. In that, first we will learn color blindness. Discovered by Horner named scientist. Discovered by Horner, and you know that color blindness is X-linked recessive disorder. It is most commonly seen in males than the females. Why? Because in females, 2 X chromosomes are there, so very less ratio is there. Here, 2 types are there. Proteinopia, that is red color. If person is unable to identify the red color known as proteinopia, another is deuteranopia, that is green color. Color blindness is checked by chart. So here I can stop now.